Hello. So we are waiting for a few more minutes to let others join if some have to, and then we can start with the class. So just a few more minutes and then we will be starting.
Okay, sir.
Hello, sir.
Yes.
Good evening, sir.
Hello.
Good evening, sir.
Good evening. Good evening. We are just waiting for a few more to join and then we will be starting. Probably we will
Oh,
be starting now only. If
okay.
others are joining, they can join later. Just few more seconds. Okay.
Till to the other joining, can I ask um,
Yes, yes, sure. Go
one
ahead.
question regarding
Yeah,
to this?
yeah,
After completing this experimental biochemistry course,
yeah.
what kind of jobs and other fields I can join to that?
Okay, so see, means just speaking of the course, this
Uh,
is just the basics of the experimental biochemistry course that we do regularly in labs and in different research centers. Okay,
uh,
so these are the basics of all experiments. So if
uh,
you ask me what kind of jobs, probably you can be a part of a say a project. Okay,
uh,
a
mm,
project intern or a project assistant, and at the same time you can also be a part of different industries. Okay,
uh,
so you can take part in different industries like we have the research associates and all. Okay,
mm,
so mostly project fellows and you say being in industries.
Okay.
Okay, and you can also be part of lab technicians and all.
Mm-hmm.
So these are the open jobs based on only these experiments, if I tell.
Ah,
And if you also, alongside this, if you are also taking up any other certificate course and specialization courses, then probably you
ah,
can go into the applications portion and
ah,
in different areas or sectors that actually deal with that. And now
ah,
a lot of options have yet opened up on different things, so you can also
mm.
try them.
Acha.
Okay.
Um, Sir, I am actually a BSc in chemistry, and after that, I have done master of in medical anthropology. And
Okay.
now I have to, one thing I desire to after this course to do a MSc course in biochemistry.
Okay. So yes, you
Uh,
can go ahead with all of these. That is not a big problem. That will
uh,
be
after
definitely helpful.
after BSc chemistry, is there any chance to be a assistant lecturer or tutor in a medical college?
Um, Nowadays, things have become much more easy. So you can, yet it has become a lot complicated. But yes, you can definitely try in some, say, uh, small colleges and all.
Hmm.
You can definitely try that. Okay, but nowadays, but if you go on to clear the CSIR exam, and then you can become a lecturer.
Acha.
Okay, so with so you take so we have CSIRs happening every year, probably
Hmm.
twice every year.
Yes.
So you can give a clear one exam and become guest lecturer, or you get the certificate for lecturership.
Okay. It means that there is a probability and chance to join
Yes,
in the
yes,
medical
surely, surely.
industry.
Always there is a chance. There is
Because the
always a chance.
MBBS courses and other medical related courses, there is a have a biochemistry. They have to on first year or second year they have one syllabus there.
See, about the medical thing and all, I do not have much idea as to about the syllabus and all, but
Uh,
so I cannot tell deeply or cannot comment deeply on that.
I have seen that their course they have their first year and second year they have a subject. They have the subject biochemist.
So yes, you can definitely try something over there as well.
Okay.
Okay, so let us start with the class then.
Uh-huh.
Yeah.
Sir, one thing, how can I be make a certificate as a gold certificate? How can I obtain in this course?
I did not get you. What are you telling?
After how can I prepare for this course so I get a gold certificate?
Okay, okay, right, right. So for this, you have to just go through the lectures thoroughly. That is
Uh-huh.
number one. Second is that whatever homework assignments are there, you have to keep solving them in a
Uh-huh.
with a good dedication and all, and try to solve the assignments on your own by finding out the formulas, the answers to the different questions and all. And
Uh-huh.
the third thing that you have to do is on a compulsory basis you have to attend these live sessions where we
Uh-huh.
discuss the previous year assignments and all. And if you are having any particular doubt and
Uh-huh.
doubts and all, so you can definitely come and ask over here. That will be definitely helpful. And lastly is that you have to, before the exam, you have to just go through and revise properly the lectures and all the assignment questions, the solutions to them. And I think that will be good enough to clear the exam with a very good score.
Also, sir.
Okay, with
Uh-huh.
solving the assignment is also having a good percentage in your final exam. So
Uh-huh.
just solving the assignments and coming to the live session such that there is a one is to one interaction between the students, and we can solve their doubts clearly. So that will be actually beneficial in the final exam, so you can see
Uh-huh.
the results in the final exam. and their questions are more or less similar to what has been given in the assignments and all, so that will be beneficial, and exams are not very tough if you revise and practice regularly.
Sir.
Okay, fine. So welcome everyone to the week 2 live session course on experimental biochemistry, which is taught by Professor Shagota Dasgupta and Professor Shoumode of IIT Kharagpur. I am Srijan Sen, and today I will be taking the week 2 live question answer session. So every time, like I do, is first I go with the brief discussion about the lecture slides that you people have already seen, and then I would be discussing with you the question answers of say previous year or important question answers that will help you in solving the assignments and taking up the exams in the near future. So these are actually beneficial for your competitive exams as well, so you can try doing that. Okay, so I am sharing my slides with you. Okay, so is my slide visible to you all?
Sir, now visible.
Okay, great. Okay. So in today's lecture, we would be talking mostly about the amino acids, the titration of amino acids, the different structure of amino acids, and then mostly we would be talking about the determination of the isoelectric point. So today's concepts will mostly deal in amino acids and their properties, followed by the isoelectric point, then electrophoresis and the titration determination of different amino acids, say charged amino acids and the neutral amino acids. So before I directly jump into the concepts of amino acids, their structure and all, so amino acids are the essential elements of life that supports the different functions of the body, because these are the building blocks of the proteins that are necessary for carrying out the different functions inside our body. And this amino acid, from the term as we understand, it is made up of an amino group and a carboxylic group, that is the acidic group, and this term is used to refer to an alpha amino carboxylic acid. So the simplest known alpha amino acid is the amino acetic acid, also known as the glycine. And there are other commonly used amino acids that have side chains substituted on the alpha carbon atom, and these when substituted with a methyl group gives us a alanine. Okay, so this is the structure of the amino acid. So the first amino acid that was discovered was in France in the year of 1806, and that was isolated from asparagus, and followed by it was named asparagine. And then later on, what happened? Cysteine was discovered from urinary stones, and glycine from gelatin, and from the muscles and wool we came across leucine. And there were a recent few scientific developments that occurred, and by the year of 1935 all the amino acids that make up our proteins had been totally discovered. So many of us we have heard of the term glutamate. So a German scientist named Karl Henrik Richthofen, okay, he isolated this glutamate from the wheat protein gluten in the year of 1866 and gave it the name glutamate. Then another scientist, Japanese researcher Kikune Ikedi, he also discovered this glutamate that gave you kombudashi, that is a stock made of kelp, in the year of 1908. And these extensively inspired the research in different countries, and it also helped to understand the functions of the amino acids. That is why today we are studying about amino acids and their essential functions in the human life. So, what are amino acids? Monomer units from which the long polypeptide chains of proteins are formed. So, these are the building blocks of your proteins. So, L-alpha amino acids and their derivatives participate in different cellular functions that deal with the nerve transmission, biosynthesis of porphyrins, purines, pyrimidines, and urea. So short polymers of amino acids are called peptides that perform prominent roles in the neuroendocrine system as hormone releasing factors, different functions related with the hormones. Then we have the neuromodulators and the neurotransmitters. Now, although there are several hundreds of amino acids found in nature, only about 20 amino acids are needed to make up all the proteins in a human body or other life forms. So we call them as the naturally occurring amino acids. And these 20 amino acids are mostly populated around the L isomer alpha amino acids, and all of these amino acids except for glycine contains a chiral alpha carbon, because as we know from the chirality concept that the four hands of the carbon atom should contain four different atoms, but your glycine has two hydrogens on the alpha carbon atom. That is why it is an achiral molecule. And all these amino acids are L isomers with an R absolute configuration except for glycine because it has no chiral center, and cysteine because of the sulfur containing R group. That is why it has an S absolute configuration. And then these uh, Okay, so there are certain microorganisms also that secrete free D amino acids or peptides that contain both the D and L alpha amino acids. Mostly, you would get to know when we speak of the bacterial cell wall. Okay, so that is made up of D-alanine, D-alanine. Okay, so there are a few amino acids that also are secreted by different microorganisms, and several of the bacterial peptides are of therapeutic value, including your antibiotics bacitracin, you have the gramicidin A, the anti-tumor agent bleomycin, etc. Now, some proteins contain additional amino acids that arise by the post-translational modifications of different amino acids. Like we have the 4-hydroxyproline that is derived from a proline. Then we have the 5-hydroxylysine by the post-translational modification on a lysine molecule. So there we have the acetylation, the methylation, formylation, and phosphorylation of certain amino acid residues, and your naturally occurring amino acid gets converted to a different amino acid, and it gives rise to a unnatural amino acid. So these modifications significantly extend the biologic diversity of proteins by altering their solubility, stability, their catalytic activity, and interaction with other proteins. So now we will talk about the structure of the different amino acids and the short codes that we use for these amino acids. So there are 20 different naturally occurring amino acids that we just spoke of, and these are the different R groups that are substituted in the amino acid structure, and then we give it 20 different names. Like we have the alanine standing for A, we have arginine as R, where we have this guanido group. Then we have asparagine, that is also the where your OH is substituted by an NH2, and the other thing is called the aspartic acid. So asparagine is N, aspartic acid is D. Then we have cysteine, glutamic acid, glutamine. We have glycine. We have the histidine. We have the isoleucine, proline, phenylalanine, methionine, lysine. We have leucine, serine, threonine, tryptophan, tyrosine, and valine. So, and when after all of these 20 amino acids were entirely discovered, they have to be separated based on something. So they were segregated based on the aliphatic side chains, their aromatic side chains, whether they are acidic, basic, or hydrophilic, whether they are sulfur containing or NH2 group containing, and on the basis of the two most important non-essential and the essentially occurring amino acids. So the red stands for aliphatic, green for aromatic, orange for acidic, blue for the sky blue for basic. Then we have pink for hydroxylic. Uh, this is your yellow for sulfur containing, the dark blue for amidic, and then the full circle for non-essential and the dotted circle for the essential amino acids. So we have all the amino acids that are segregated on the basis of such different categories, and what we need to keep in mind is the structure of these amino acids, because whenever you are in a structural biology lab or a lab that deals with the protein structure and the amino acids, these structures will play different roles. Now speaking of the essential amino acids, we have isoleucine, leucine, valine, then phenylalanine, tryptophan, we have histidine, methionine, lysine, and threonine. The others fall in the category of non-essential amino acids. So just from the structure, we have a few questions, like a pair of amino acids containing non-polar and aliphatic side chains. So there are four options that are given. So what will be the answer? Kindly let me know. So the pair of amino acids containing non-polar and aliphatic side chains. Anyone who can give me the answer of this? The pair of amino acids containing non-polar and aliphatic side chains.
Methionine, alanine, and leucine.
Yes, that is the correct answer. Option B, alanine and leucine. Okay, because this alanine is non-polar and the aliphatic side chain is present in leucine. So yes, that is the correct answer. Okay, and the next one, which of the following is an amino acid? So you can just type the answer in the chat as well. If you're not comfortable speaking, that is fine, but it would be good if you are also answering. You can just,
Yeah, no, no, no. I'm only on the mobile doing so that's why.
no, no, it's okay, it's okay, it's fine, absolutely fine. So so what will be the answer for this? Which of the following is an amino acid? Nusrat, would you like to give it a try? No.
So second one.
That is proline, right?
Yeah.
Okay, that is correct. So yes, proline is an amino acid, and your non-polar and aliphatic options are correct. So that is good. Okay, the next one. Number of chiral centers in isoleucine and leucine are. So now we have to tell the number of chiral centers in isoleucine and leucine. Okay, for the time being, I am getting back to the structure of these amino acids, and then you can give me an answer. Okay, so we have isoleucine and leucine over here. So tell me, how many chiral centers does isoleucine and leucine have? So chiral center means the particular carbon atom should have four different groups in its four hands. So that will be the chiral carbon atom. Okay, so any answer? So how many chiral carbon does isoleucine have, and how many chiral carbon does leucine have?
Sir, 4?
Four. Four for what? Means that's the total you are telling.
Yes, sir.
No, no. Tell me how many chiral carbon atoms does isoleucine have, and then tell me how many does leucine have. So 1, 2, 2, 1, 2, and leucine is achiral. Both are achiral. So you have 1 and 2, 2 and 1, 2 and 0, and both are 0. So what is your answer now? So those are the options. Okay, let us see. So if you check, this is isoleucine and this is your leucine. So if I try to draw the carbon atoms that are chiral, so this one is definitely chiral, we all know, because it has a carboxylic group, NH2 group, hydrogen, and this big group. This is also chiral carbon, we know. Okay, so for the same reason. Now the next one is we are looking into this carbon. So if you look into this carbon has two hydrogens. So this is not the chiral center. And also if you look into this carbon, it has two same methyl groups. So that's why this is also the chiral carbon. So leucine has only one. And if we look over here, this is the another carbon atom that is also chiral. So 1 and 2, because it has a methyl group, it has an ethyl group, it has another group and an hydrogen. So that is why 2 and 1 is the correct answer. Okay. Okay. The next question that I have is tyrosine and tryptophan are less hydrophobic than phenylalanine. Why? Why is tyrosine and tryptophan less hydrophobic than phenylalanine? So which of the option is correct? One is phenyl side chain has a phenol group in it, has no polar groups attached to it. Tyrosine and tryptophan have similar side chains, and phenyl side chain has indole group attached to it. So which of the following option do you think is correct? So from the structure, if we see that their tyrosine and tryptophan have phenolic group and indole ring respectively. Okay, so the correct answer would be that tyrosine and tryptophan are less hydrophobic because, okay, let's go from the structure and just see it once. So see, we have tyrosine that has an OH group attached over here. We have a tryptophan and it has a carboxylic group over here. But on the other hand, what about phenylalanine? So phenylalanine, where is phenylalanine? Yes, phenylalanine has this particular group. That is, it does not have any uh, hydrophobic or polar group that is attached to this portion. but your tyrosine has an OH group and your tryptophan has a what group? Tyrosine has a phenolic group and your tryptophan has an indole group. So they are less hydrophobic because phenyl side chain does not have any polar groups that is attached to it. So B is the right option over here. Here we have also B as the correct option, and here also B, and this is also B. Okay, let's move on now. So moving on to the next type of an amino acid, that is the 21st amino acid, and there can be even more, 21st, 22nd, all. But what we consider as the 21st amino acid is that this is the selenocysteine. So we have the cysteine over here, and this is the selenocysteine. Nusrat, can you mute your mic? Yeah, thank you. Right. So the 21st amino acid, that is the selenocysteine. So humans contain approximately 2 dozen selenocysteine that include different peroxidases and reductases. That is the selenoprotein P, which circulates in the plasma. Now, peptidyl selenocysteine is not a product of the post-translational modification, but it is inserted directly into the growing polypeptide during a translation. However, unlike the other 20 protein amino acids, incorporation of selenocysteine is specified by a large complex genetic element for the unusual tRNA or tRNA coding for selenocysteine. So we have classified amino acids based on non-polar hydrophobic, polar hydrophilic, and then categorizing them on the basis of aliphatic aromatic residues and acidic neutral and basic residues. So all 20 amino acids differ in chemical structure, volume and shape. At room temperature, all the 20 amino acids are crystalline solids. They are water soluble, they are colorless, and at neutral pH, these amino acids exist as zwitterionic in their zwitterionic form. So zwitterions is that they have a neutral charge with the positive charge on the amino group and the negative charge on the carboxylic group. So they have a neutral charge. Now, polar amino acids. So we have 10 amino acids that are polar, like the acidic amino acids have aspartic acid and glutamic acid. In basic, we have histidine, lysine, arginine. Then in neutral, we have cysteine, asparagine, glutamine, serine, and threonine. We mostly need to keep the names and the structures of these amino acids in mind, because there will be a few questions based on the structure and the type of amino acids. So then we categorize them on hydrophilic and hydrophobic aspects. So the polarity of an amino acid depends on the side chain. So polar amino acids make hydrogen bonds with suitable groups like OH, NH2. Okay, and these polarity of amino acids affects the overall structure and function of proteins like sickle cell anemia. Okay, so just by a single nucleotide mutation, one amino acid changes to the other form, and that causes a diseased condition. So now we are looking from the perspective of amino acids, but later on these amino acids will join together via peptide bonds to give us proteins. So we have to always look as to how is it going to affect the human body and the dysfunctioning of different proteins. Okay, so that is why even if there is a particular mutation in the single structure of an amino acid, or it gets mutated to a different amino acid, the entire function of the protein can be disrupted, and that can lead to disbalance and cause different kind of diseases. So essential amino acids. So humans can synthesize about half of the amino acids needed to make proteins. Other amino acids that the humans cannot synthesize are known as essential amino acids that must be provided in the diet. So we have 10 essential amino acids that I've mentioned before, and proteins that provide all the amino acids in the right proportions are called complete proteins, like meat, fish, milk, and eggs. And about 50 gram of complete protein per day is necessary for the adequate is necessary for adult humans. And there are several proteins that severely are deficient in one or more of the essential amino acids, and these are called incomplete proteins. Now, if the protein in a person's diet comes mostly from incomplete source, the amount of human protein that can be synthesized is limited by the amounts of deficient amino acids. So these are just a few informations on the essential amino acids. Also, we have rare and unusual amino acids that derive from the post-translational modifications. Like we have the hydroxy proline, 5-hydroxy lysine, and these are the hydroxylated versions of your proline and lysine respectively, and we call them as the rare amino acids, even though they are commonly found in collagen. So some of the less common D-enantiomers of amino acids are found in nature. Like I was mentioning, D-glutamic acid found in the cell walls of many bacteria, and D-serine is found in earthworm. Now we have naturally occurring amino acids are not alpha amino acids, like we have the gamma GABA, that is one of the neurotransmitters in the brain, and we have beta alanine that is also a constituent of the pantothenic acid. Okay, it's also the cofactor for some vitamin. Now we will be speaking about the acid-base properties of amino acids. Now this is very important because amino acids are, after all, charged molecules, and they can have also charge groups in their R position. So that will actually make these amino acids very sensitive to different changes in the pH. Although we write the amino acids in the uncharged form, but their actual structure is ionic and it depends on the pH. So the carboxyl group loses a proton and gives the carboxylate ion, and the amino acid group is protonated to an ammonium ion, and this structure is called the dipolar ion or the zwitterionic form. So the dipolar nature of amino acids gives several unusual properties. That is, amino acids have very high melting points over 200 degrees centigrade. That is, glycine has about a melting point of 262 degrees Celsius. And amino acids are more soluble in water than in organic solvents. Amino acids have large dipole moments than the simple amines or simple acids. From the example, you can see glycine has a dipole moment of 14D. Propylamine has a mu of 1.4D, and propanoic acid has 1.7D. So it's definitely high for amino acids. And amino acids are less acidic than carboxylic acids and less basic than most amines. So in fact, the acidic part of the amino acid molecule is the NH3 plus group and not the COOH group, and the basic part is the carboxylate group and not the amine group. Okay, now because these amino acids are both acidic and basic in nature, so we call them as the amphoteric nature or property, having both acidic and basic properties. Now the predominant form depends on the pH of the solution. In the acidic group, the group is protonated to a free group and the molecule has a positive charge, and when we raise the pH, the proton is lost at about pH 2, and this point is called the first acid dissociation constant. Now when we rise the pH even higher, the group sorry, yes. So when we rise the pH, what happens is that the the NH3 plus group loses the proton, and we get the neutral molecule, and at about pH 9 or 10, and this point is the second acid dissociation constant. So above this pH the molecule has a negative charge. So in the first stage it's cationic in acid, and on losing the proton is lost from the carboxylic group, and after that it is lost from the NH3 plus group, and then at very high pH the amino acid is negatively charged. So, when we speak of the amino acids, acidic and basic properties, the very important concept that we need to know is about the isoelectric point. So, isoelectric point is the point where the amino acid is neutral or it is in the zwitterionic form. So an amino acid bears a positive charge in acidic solution and a negative charge in basic solution. So there must be an intermediate pH where the amino acid is balanced between the two forms and where it is neutral. So we call that pH as we call that form of an amino acid as a zwitterionic form, and that pH as the isoelectric pH or the isoelectric point. Like the side chain of aspartic acid and glutamic acid contains acidic carboxylic group. So these amino acids have acidic isoelectric points at around 3. And an acidic solution is needed to prevent the deprotonation of the second carboxylic acid group and to keep the amino acid in its neutral isoelectric state. Whereas when we look from the perspective of a basic amino acid, like we have lysine, histidine and arginine, so they have pH values of 7.6, 9.7 and 10.8 respectively. So these values reflect the weak basicity of your imidazole ring and the intermediate basicity of an amino acid group, and also the strong basicity of the guanido group that is occurring for the arginine. And the basic solution is needed to prevent the protonation of the basic side chain to keep the amino acid electrically neutral. Now the other amino acids are considered neutral with no strong acidic or basic side chain. So their isoelectric points are slightly acidic because of the NH3 group is slightly more acidic, and the COO minus group, that is the carboxylate group, is more basic. Up to this much it is okay with the concept of acid and bases. So if you have any questions up to this, you can ask me. Okay, so I guess there are no questions. So let us proceed with the next concept, where we would be speaking about the titration of the amino acids and the electrophoresis study. So what is electrophoresis? It uses the differences in the isoelectric points to separate the mixture of amino acids. Now is a streak of amino acid mixture that is placed in the center of an acrylamide gel or a piece of filter paper with a buffer solution. Now there are two electrodes that are placed at the two edges of the gel or paper, and we apply the voltage. Now positively charged amino acids will be attracted towards the cathode, and the negatively charged amino acids will be attracted towards the anode. And amino acid with no charge, that is neutral at the isoelectric point or that pH of the buffer, will not move. So if we see from here, like we have a mixture of alanine, lysine and aspartic acid in a buffer solution of pH 6. So alanine at its isoelectric point and its zwitterionic form is at a net charge of 0, A pH of 6 is more acidic than the isoelectric point of lysine, so lysine is in the cationic form. And aspartic acid that has an isoelectric point of 2.8 is in an anionic form. Okay, so we need to keep one thing in mind: when the pH is less than pi, okay, when the pH of the buffer is less than the pH of than the pi of the amino acid will be positively charged. So pH less than pi equals to positive charge, and pH greater than pi equals to negative charge. That is why aspartic acid has a negative charge and lysine has a positive charge. And on the basis of that, at a buffer solution of pH 6, your lysine will move towards the cathode, aspartic acid will move towards the anode, and alanine will not show any movement. Okay, so so now how do you get all these amino acids? So after a period of time, when the amino acids are separated, they are recovered by cutting the paper or getting the gel, getting the bands out of the gel, and then we can treat it using ninhydrin to make the bands visible. And these amino acids are identified by comparing their positions at through by using different standards. So this is the very crude method, and we can do it in the lab by just a power supply and by using a particular buffer, and then separate them out. And in this way we can also separate the different positively charged as well as the negatively charged amino acids by using the buffer in that manner. For example, if something has a if some amino acid has a pH of say 2 and the other has a pH of say 3.6, so if we use a buffer, but if we are using a buffer of pH 6, it will not affect, because both will move towards the anode. Okay, but if we are using a buffer of pH say 2.8, then it will show movement in two different directions. Also at the same time, even if we are using a buffer of pH 6, they might be moving towards the anode, but their movement will be in different places. So the amino acid with a pi of say 3.6 or a pi that is close to the buffer range will move slowly, and that with a pH of pi of 2 will move fast. So in that manner also we can separate them out. Okay, now we will be speaking about the titration curve of glycine. So mostly we would be focusing on the titration curve of a neutral amino acid and the titration curve of a charged amino acid, and then we can jump into the questions. So first let's speak about the titration of the glycine. So glycine, as we know, is a neutral molecule, but at very low pH, that is when we start the curve from the bottom left, it is cationic, because it is far below the first isoelectric point. So now we add NaOH or a base slowly, and then we record the pH. So at a pH of 2.3, we have half of the cationic form is converted to the zwitterionic form. So below this pH of 2.3, glycine is positively charged. At pH 2.3, we have an equal population of the cationic form as well as the zwitterionic form. Now as we add more and more amount of base, the cationic form starts decreasing and the zwitterionic form increases. At isoelectric point, that is the point where the zwitterionic, all the glycine is in the zwitterionic form. That is the isoelectric point of a particular amino acid. Now, on adding more and more amount of base, and when the pH reaches 9.6, half of the zwitterionic form is converted to the basic form, that is the negatively charged form. So here we have positive, here half positive, half negative, half neutral. Here we have only neutral molecules, that is the zwitterionic form. Over here we have half zwitterionic form and half negative molecules, and above this pH we have mostly the population of the negative form. So from this graph we can see that the glycine is mostly cationic at pH below 2.3, zwitterionic at pH between 2.3 and 9.6, and anionic at pH above 9.6. And by varying the pH of the solution, we can control the charge on the molecule and separate them on the basis of electrophoresis by using different buffer of different pH to separate them out. And the next one is when we have a charged molecule. Say for glutamate we have a negatively charged side group, and for arginine we have the positively charged side group. So the concept remains the same. In the beginning, that is in the beginning we have the positively charged, where the charge of the molecule is +1. Then when we reach over here, that is at pH 2.19, here we have equal population of this positive charge, that is the positively charged glutamate, and the zwitterionic form, that is where the charge is neutral, that is 0. Then when we start adding more of base below pH 4.25, or at pH 2.45, we have equal contribution of the zwitterionic form as well as the negatively charged molecule. And when we reach 9.67 pH, you have equal contribution of this negative -1 charged molecule and the -2 charged molecule. So we have two negatively charged groups over here. Here we have two negative and one positive. So the charge on the form is -1. Here we have -2. Here we have 0, and here we have +1. So we start with +1, 0, -1, and -2. So in the beginning it is +1. At pH 2.19 it is +1 and 0 in equal population. At pH 4.25 it is 0 and -1. At pH 9.67 it is -1 and -2, and above this pH it is only -2. Whereas on the reverse, on a different, for the positively charged, in the beginning we have plus, at below pH 2 we have +2. At below pH 2 we have the charge on the arginine is +2. Then at pH about 8 we have +1, 2 positive, 1 negative, so +1. At pH about 10 the charge is 0, and at pH above 13 the charge is -1. So for positively charged groups we move from +2, +1, 0, and -1, and for the negatively charged groups we move from +1, 0, -1, and -2. And on the basis of that we can calculate the PI. So how do we calculate the PI? So we have to consider two pK values that lies between the -1 and +1 forms. So here we have here we have +1, 0, -1, -2. So +1, 0, -1. So we have to consider the pKs between -1 and 0 plus the pK between 0 and +1 divided by 2. Same way for arginine we will be doing. We have +2, +1, 0, and -1. So PI will be calculated by the pK between these two, that is 9 plus 12.5, because it is between -1 and 0, and this is between +1 and 0. So 9 plus 12.5 divided by 2, that is coming about 10.8. For glutamate we have the pK1 as 2.19, the pKr as 4.28. So these two divided by 2, so we have 6.4 divided by 2, that is around 3.23. So in this manner we can find out the PI of all the amino acids, whether they are neutral, positive, or negatively charged. Only what we need to keep in mind is the charge on the particular amino acids, and the charge on the side groups. So the first uh, proton is always lost from the carboxylic group, and then it is lost from the amine group, and then from the side chains. Okay, or in some cases it is first the side chain and then the carboxyl, the amine group. Okay, so if we get to know about the charge on each of the forms at different pH, we can easily calculate the pi of the particular amino acids. Okay, and for glycine it is very simple. It is only the, we have 2 pk, so we add the 2 pks and divide by 2. So 2.3 plus 9.6 divided by 2, that is 12 by 2, so 6. So this is simple for uncharged amino acid, but for charged amino acid it becomes a little bit tricky. But if we find out the charge on the particular forms of that amino acid in different pH, we can do it easily. Okay, so that much we have for our discussion portion. Next I would be solving a few questions with you people. So up to this, if you have any questions, you can ask me. Up to this much, if you have any doubts on any portion.
Sir, can you explain this slide again?
This slide?
Yes, sir.
Okay, sure. So let us see. Okay, if we talk about a charged amino acid, let us first find out what is the charge. So at pH 0, the amino acid will be positively charged. Why is it so? Because there is only HCl that is present, and it is in the acidic solution. So when it is in the acidic solution, all the ionic groups will be protonated. So when everything is protonated, this amide group will also be protonated, and the charge on the molecule will be +1. Okay, now when the pH just rises above 2.19, so when we go to a pH above 2.19, what will happen is that the hydrogen from the carboxylic group, so this pK1 is for the hydrogen in the carboxylic group, so this proton is lost. Okay, so when this proton is lost, the charge on this amino acid becomes 0, so it is neutral or zwitterionic. Okay, what happens now when when the pH rises over 4.25? So this pK is for the side chain. Okay, so pKr, that is R stands for the side chain or the R group. So this pK is for the side chain carboxylic group. So above this pH, the proton will be lost from the side chain carboxylic group. And when the proton is lost from the side chain carboxylic group, what do we have? We have a positive charge over here, we have negative over here and negative over here. So -1, -1 and +1. So we have -1. And when the pH is, so when we add NaOH and the pH rises over 9.67, so all the protons are deprotonated, all the groups are deprotonated, and the proton is lost from this NH3 plus group, and we get the charge on the molecule to be -2, because minus minus. So first it is lost from the main carboxylic acid group. Yes, so first it is lost from this one, then the hydrogen is lost from the side chain group, then it is lost from the amide group, and finally we get this particular form. Okay, this much is clear?
Yes, sir.
Okay, and now the same thing happens for your arginine as well. So what happens? So okay, so when the pH is below 2, that time all the groups are positively charged, and arginine has two NH, two groups, okay, amide groups. So both these amide groups will be charged. So the charge over here is +2. Now, when the pH is close to your 8 or say 9, okay, so when it is close to 9, one hydrogen will be lost. And what is the hydrogen that will be, so when the pH is above 2.1, at that time your hydrogen will be lost from the carboxylic group. So it has only one carboxylic group, so that hydrogen will be lost. So we have +2 -1. So the charge is +1. Next, what happens? So next, what happens is that when the pH rises over 9, so when the pH rises over 9, the proton is lost from the backbone amide. So when the pH is when the proton is lost from the backbone amide, the charge is 0. Okay, because +1 -1. And finally, above pH 12.5, what happens? The positive charge is lost from this guanido group, and the molecule becomes negatively charged, that is -1, because of the negative charge of the carboxylic group. So at extreme low pH, the ionic groups will be positively charged. At extreme high pH, the ionic groups will be negatively charged. And on the basis of that, we can talk about the charge. Okay, now we know so we have to take the pKs. Okay, so the pK, only those pKs will be considered that is present between +1, 0, -1. Okay, not -1, -2, -2, -3, nothing like that. Only +1 to 0, that pK will be considered, and 0 to -1, that pK will be considered. Same over here, -1 to 0, that pK will be considered, and 0 to +1, that pK will be considered. So what do we have? We have 12.5 and 9, that is over here 12.5, over here 9. So 9 plus 12.5 divided by 2, 10.8. Over here we have 2.19 and 4.25. Not this one, we will not consider 9.67, divided by 2, that is 3.23. Okay. And also another way you can keep this thing in mind, we have to, for calculating PI, we have to consider those two pKa's that are very close to each other. Like 2.194, 9 is very far, we will not consider it. So 2 and 4 are close, so add them, divide by 2. Here 2 is very far from 9 and 12.5, so 9 and 12.5, add them, divide by 2, 10.8. As simple as that. Okay, clear now?
Yes, sir. Thank you, sir.
Good. Okay. So let us take 5 minutes break, and then we can start with the practice sessions. Okay. Okay, so let us start with the practice problems. So the first question is the pair of amino acids containing aromatic side chains. So which pair of amino acids contain the aromatic side chains? And these are the type of questions you will be expecting even in your final exam. Might be easy, but if you get trapped by the structures and all, if you are not aware of the structures and all, you might get trapped. So these are actually easy questions. You can easily answer them and score a good marks. Okay, so the pair of amino acids containing aromatic side chains. You can write in the chat box if you want answer. So we have a few amino acids that are responsible for the absorbance of the particular protein molecule when these amino acids are present. So among them we have phenylalanine, tyrosine and tryptophan. So option A is correct. Yes, very good. Okay. The next one, just right. The next one, the pair of amino acids, both of which are expected to have a net positive charge at pH 7.4. So we have a buffer of pH 7.4, and which of the amino acids will have a positive charge at pH 7.4? So as I've spoken, when the pI is less than pH, when the pH is less than your pi, it will be positively charged. When the pH is more than your pi, it will be negatively charged. So I have given you the hint. Now give me the answer. So the pi of the amino acids have to be more than the pH. So which pair of amino acids have pi more than 7.4? So a pH of 7, a pi of pH 7.4, above 7.4 means a pi above 7.4 means they are basic amino acids. So which basic amino acids do we know? We have histidine, lysine, arginine. So these are the short forms, but probably even in your question paper there might be questions like the one letter code. Okay, like for lysine we have K, for arginine we have R. So you should be aware of these one letter codes as well. Otherwise you know the answer but you cannot answer it. Okay, so lysine and arginine will be positively charged at pH 7.4. The next one, which of the following statement is or are true for isoelectric point? So now we have to find out what is an isoelectric point. The pH where the molecule carries no net charge, the pH where the carboxylic group is uncharged, the pH of maximum electrolytic mobility and field point. Very good. So yes, the pH where the molecule carries no electric charge is known as the isoelectric point or the isoelectric pH. Okay, let's go to the next one. Which of the following pairs have imidazole and indole rings in its structure? So first is the imidazole ring and the next one is the indole ring. Very recently I have spoken about it. So we have an indole ring and we have an imidazole ring. So indole ring I recently spoke where we have tryptophan. So tryptophan has the indole ring. Okay, might be you're not aware of the structures at present, but for your exam, kindly keep in mind the structure of the amino acids. Very easy questions are going to be there from these portions from the first few lectures. There are going to be very easy questions. There is nothing to trick the questions about, just that you have to keep things in mind. If you can keep at least a few of the at least not all the structures in mind, you can get sure shot questions and you can score good. So imidazole is so histidine has an imidazole ring and tryptophan has an indole ring. Also, just another thing that there can be like which amino acid has a there was a question for proline, okay, as an imine group. Also, which amino acid has a guanido group? Then you have to answer it's arginine. Okay, so there can be a few questions like that. Okay, the next one. Which of the following has or have the most chiral centers? Isoleucine, serine, tryptophan, threonine. So which has the most chiral centers? Okay, I would take that, but alongside that, so this has 2, this has 1, tryptophan has 1, and what about threonine? Threonine also has 2. So the correct answer is 1 and 4. Okay, let's see about threonine. So where is the structure of threonine? Okay, so here we have threonine. So this is the one, two. So this carbon will have hydrogen, methyl, OH and this big group, and this carbon will have this group H, amide and the carboxylic acid. Okay, so it is different. But for serine, we have 2 hydrogens over here. So wrong. And for tryptophan, we have only this group has 2 hydrogens and all the other carbons are achiral. So D is the correct option. So 2, 1, 1, 2. So 1 and 4 will have most chiral centers. Okay, the next one. Calculate the pi of the amino acid where pK1 is 2.16, pK2 is 9.06 and pKr is 10.54. So we have this group and we have to calculate the pi of this group. So what is the pi? So what is the pi of the particular amino acid?
Five point.
So you are saying 5.61?
This is
Okay, let me see what answer Nusrat gives and then I would be telling you.
PK. PI value.
Yeah, the pi value.
PK1 plus PK2 divided by 2.
If you see, okay, so if you see from over here, what I said is where the charge is +1 and where the charge is -1. So we have to consider the pKs between that. And also what I mentioned is that the two pKs that are close to each other will be considered. That is a short way, but what is the scientific concept? The concept is where we have to consider the middle point to be the zwitterion and we have to take the pK before that and the pK after that. So if you see over here, where is it? Right. So over here, the first pK is where it has a charge of +2.
Hmm.
Sorry, +1. Then uh, no, wait. So below this it is +2, and after this, so this is lysine. It will have a similar kind of thing like arginine. Okay. So where we have a +2, then we have a +1 above pH of 2.1, and then above that we have 0 and -1. So here we consider 9 and 12.5 because it is between +1, 0 and -1. So here also we have to do the same. So +2, +1, 0 and -1. Yes, -1. So pK2 is 9.06 and pKr is 10.54. So 9.06 plus 10.54 divided by 2, that is 9.8. Okay, clear? Right. Okay, let's move to the next question. Which of the following is or are a set of polar amino acids? So, which of the options um, speak of polar amino acids? So, polar amino acids means it has an ability to interact with the water molecule, or it has polar side groups. So, which of these are correct? See, here questions are all from the structure and about the amino acids. So, it won't be tough. Okay, just you have to keep again, the structure of the amino acids in mind. So, mostly these are from the structures. So if you see, serine has a OH group, threonine has an OH group, and aspartic acid has a COOH group. Here we have aspartic acid, asparagine, and threonine. So here both C and D would be the correct option because all of these have polar side chains. Now the next question. When amino acid alanine is added to a solution with a pH 7.3, alanine becomes. So what would be the charge on alanine at pH 7.3? So if you can remember, it has a pi of 6. So if it has a pH of around a pi of around 6, what would be the charge on on alanine at pH 7.3? So here pH is more
Negatively
than,
charged.
right, very good. So it will be negatively charged because pH greater than pi negatively charged, pH less than pi positively charged. Okay. Now when NaOH is added to a solution of lysine dropwise, how many inflection points do you observe? So inflection points are the points where there are sharp changes. Okay, so when you are adding NaOH, how many sharp changes will you observe when in the titration of lysine? So the hint over here that I would be giving is the number of pK is it has -1. So number of pK is it has this particular molecule -1. So that would be the number of inflection points. Now tell me. So the sharp changes in pH. Where do you observe sharp changes in pH for lysine? So the first one would be from +2 to +1. The second one would be between, so the first pKa would be between +2 to +1. Another one would be between +1 and 0, and the third one would be 0 to +1. Sorry, -1. So the first change you would be observing, like over here, is one change and the second change. So first one over between pK1 and pK2, and the second one between pK2 and pK3. So we would be observing two sharp changes. So if we have 3 pKs, it will be 3 minus 1. If there are 4 pKs, 4 minus 1. So 3 sharp changes. And for glycine or neutral molecules, there is only one sharp change. The next one. Which of these amino acids have pI close to the physiological pH? So physiological pH means pH of blood. So which of these amino acids have pH close to the pH of blood? So the pH of blood is around 7.4. So the amino acid which has a pH close to pH 7.4, and that is your histidine. Remember that. Okay. The next one. The pI of an amino acid has pK1 2.14 and pK2 9.73. So what would be the pI? This is an easy question. So the first pK is given and the second pK is given. So what would be the pI?
So D.
Yes, D is correct. Okay. So 2.14 plus 7.93 divided by 2, so 5.935. Correct. Okay. The next one. During pH metric titration of glycine with NaOH, certain amount of HCl is added at the beginning. So before we start the titration, lot of HCl is given at the beginning. Why is it so? To neutralize NaOH, neutralize glycine, completely protonate glycine, and convert glycine to zwitterionic form.
So D.
So, sorry, I did not get you.
D.
D converts glycine to zwitterionic form. Right? You are telling B or D?
D, sir.
D. Okay. No, that is not correct. The correct answer over here is C. Why is it so? See, when glycine is taken in water or at a neutral pH, it is present in the zwitterionic form. But to carry out the titration, we have to add HCl. Why? When it is in the zwitterionic form, it is already at a pH 7. So we cannot observe the sharp change at pH below that. So when we start from pH 0, so there is nothing called pH 0 as of such, but we add the because at present in your glycine would be present in the zwitterionic form. Now we add HCl to lower the pH below 2 and start from the point 0. So to start from the point 0, we add HCl such that all the molecules of glycine are positively charged, and when all the molecules are positively charged, we can start from the point 0 and add drop by drop of NaOH and raise the pH, and then we can check the different points and calculate the pI, and also observe where there is a sharp change in the pH. So that point would be the pI of that particular amino acid. So that is why we add in the beginning good amount of HCl to actually protonate all the glycine molecules. Okay.
Okay, sir.
The next one is the pK2 for the side chain of histidine is 6 when it is present in isolated form. Considering a specific protein with histidine at its active site, the pK2 is expected to be. So what would be the specific, what would be the pK2 of histidine B when it is present in a protein? Knowing that the pK2 of the histidine, that is the pK2 that is responsible for the side chain, is 6. So what will happen to the pK2 of the histidine molecule or amino acid in the protein? So it will be less than 6 if active site has more positive charge, greater than 6 if it has more positive charge, less than 6 if it has more negative charge, and same like isolated histidine. So isolated histidine versus histidine in a protein molecule. Okay, so let us discuss this one. Now, when histidine is present in an active site where we have mostly positive charges, so what will happen? The pK2 becomes lower than 6. Why is it so? Because the positively charged in the active site will destabilize the positively charged histidine side chain. So a higher concentration of protons, that is lower pH, will be required to protonate the histidine side chain, resulting in lower pKa. So when we have the positive charge, the pK2 decreases. When we have negative charge, the pK2 increases. So this can be another kind of question in your exam, like you are given 4 options at pK at less than 6 when more positive charge, less than 6 when more negative charge, greater than 6 when more positive charge, greater than 6 when less negative charge, and 2 options can be correct. Okay, so for the time being, option A is correct, where the pK decreases because of the destabilizing positively charged around it, and that is why more hydrogen atoms would be required to protonate it, and that is reducing the pH of the pK of the particular side chain. Okay, so it becomes less than 6 if it has more negative charge, and if there were negative charges, it would be greater than 6. Okay, the next one, the correct 1 letter code for the tetrapeptide. So we have a tetrapeptide, tryptophan, asparagine, tyrosine, and lysine. So tryptophan, asparagine, tyrosine, and lysine. So what is the 1 letter code for the tetrapeptide? Is it YDWK, WATL, WNYK, or TNYL? What is it? Any idea, anyone? What will be the answer of this? So if you see the chart of my amino acid, we have tryptophan as W, asparagine as N, tyrosine as Y, lysine as K. So WNYK, where D is aspartic acid, A is alanine, L is leucine, T is threonine, and okay, so that's all we have now. So W tryptophan, N asparagine, Y tyrosine, and K is lysine. Okay. The last question for the day. Titration of arginine residue present in a protein is difficult to carry out with NaOH, unlike individual arginine. So why is it difficult to titrate arginine residue in a protein than an individual arginine? Why is it so? So the options are: protein forms complex with NaOH, multiple amino acids make the process complex, the structure of the protein may degrade at higher pH, or all of the above. So which is the correct option, or which are the correct options, or all of these? So what do you think will be the correct option? So see, when we compare any amino acid on an individual basis versus something when it is present in a particular complex molecule, so we have to also understand that it is not only the arginine that we are titrating, there are other amino acids also present alongside it. So the first thing is that at high pH or at extreme pH conditions, the structure of the protein will degrade. Okay, so that is the correct option. Okay, so the structure of the protein will degrade at higher pH. Also, there are other amino acids that will also take part in the process, thereby complicating the situation, and we cannot estimate the PI, or we can perform the pH and tell that we have done it only for arginine, because there are other amino acids also taking part in the process, and also at extreme pH conditions the structure of the protein gets disturbed or degraded, and the entire protein opens up, and then it becomes a more poor situation to carry out. So the two options are correct. That is, multiple amino acids make the process complex, and the structure of the protein degrades at higher pH. Okay. So that's all from my side. If you have any questions, you can ask me. Else we can end the class. So these type of questions you can expect in any competitive exam, not only for the experimental biochemistry NPTEL course exam. There can be all in any competitive exam. This type of questions are very common, based on the structure of amino acids, the function of amino acids, calculating the PI. Uh, then there can be questions on the charge on the amino acid at a particular pH. So there can be different, they can do different tricks to questioning. But if you know the structure, and if you have a great idea about the types of amino acids, you can easily answer them, because these are direct questions and you can try answering them very easily. Okay, so I think that's all from my side. Thank you, and I guess there are no more questions.
Thank you, sir.
Okay, thank you all, and then we will again meet in the next week,
Hello, sir.
same
Hello.
time. Yes,
Hello. Hello.
yes, tell me, I can hear you.
Sir, the assignment, previous year assignments are not uploaded yet regarding to experimental biochemistry.
No, previous year assignments won't be shared with you people. It is that we will be discussing.
Okay.
Okay, and when we will be sharing the slides, there the previous year assignments will be present in the
Okay, sir.
question answer format. So you can check back from them when the NPTEL team would be sharing with you. Apart
Okay.
from that, no other previous year questions would be shared as of
Ah,
the rules.
Okay.
Okay. Okay. Good.
Thank you.
So thank you. We end the class over here. We will
Hmm.
again meet the next week at 6 o'clock.